Springer, 2022. Rewritten in easy-to-understand language while keeping all important information for medical and dyslexic students. Part 2. 1. Polyetail, stability and translation. The polyetail is a long stretch of adenine nucleotides added to the 3' end of an mRNA molecule. It plays two vital roles. mRNA stability. The polyetail protects mRNA from degradation. Shortening by adenolases marks the transcript for decay. Translation efficiency. It interacts with polymer-binding proteins PAPs, which circularize the mRNA, facilitating the reinitiation of translation by bringing the 5' and the 3' into proximity. Key insights: optimal length, approximately 60% as for most cells, up to 120 for dendritic cells, and up to 300 for T cells. In synthetic mRNA, added by template or polymerase. Segmented polymer with minimal non-A bases improves manufacturability without major loss in function. 2. Codon optimization and secondary structure. Codon optimization involves adjusting the coding sequence to use the host's preferred codons without altering the amino acid sequence. Purpose: to increase translation efficiency and protein expression up to 1,000-fold. Mechanism: the 5' end of the ORF is kept less structured to allow smooth ribosome entry. The body of the ORF benefits from higher RNA structure, slowing translation and helping proper protein folding. mRNA secondary structures like hairpins can impede translation. Modified nucleotides such as monepsi and 1-methylpseudouridine can stabilize or destabilize these structures depending on the context. 3. Modified nucleotides, immune modulation and efficiency. To avoid excessive immune activation and ensure efficient translation, synthetic mRNA uses modified nucleotides. Modified nucleotides block detection by pattern recognition receptors, e.g., TLR378, RIG-I, MDA5, which would otherwise trigger inflammation. They also prevent antiviral enzymes like PKR, which blocks translation, and OAS, NAS-L, which destroys RNA. 4. Mechanism of action, how mRNA vaccines work. 1. Delivery. mRNA is usually packaged in lipid nanoparticles LNPs and delivered via intramuscular IM or intradermal ID injection ID. Delivery engages more dendritic cells DCS, producing a stronger immune response. 2. Uptake and expression. Cells take up LNPs via endocytosis. After escape from the endosome, mRNA is translated in the cytoplasm into the antigen protein. 3. Immune activation. Intracellular sensors like TLR7/8 detect the synthetic RNA, activating the innate immune response. APCs such as DCS and macrophages produce cytokines IL-6, TNF, IL-1 beta, and migrate to lymph nodes, presenting antigens to T and B cells. 4. Adaptive immunity. B cells make antibodies, T cells develop cytotoxic and helper responses. The immune memory formed protects against real infection. 5. Balance of immunogenicity. Unlike traditional vaccines, mRNA vaccines do not need adjuvants. The RNA itself serves as a natural immune stimulator, but this must be balanced. Too much innate response blocks translation and causes inflammation. Too little response fails to activate adaptive immunity. Hence, using modified nucleotides and purified transcripts helps fine-tune this balance.